Hey guys, welcome back to my channel Biomics. In this today's video, we are going to discuss about the 3' polyadenylation. So it is one type of modification of the mRNA. In the last video, we have already discussed about the 5' capping. So in this video, we are going to discuss about the 3' polyadenylation. So what is the requirement of this 3' polyadenylation? It is required for the transfer of the mRNA into the cytoplasm so that there will be the formation of proteins in the cytoplasm. So what happens in the DNA? There is a specific sequence which is known as poly-A signal sequence. It is present upstream of the cleavage site. So before the cleavage or before the termination, there is a sequence which is known as poly-A sequence, and it is being recognized by CPSF, which is known as cleavage polyadenylation stimulation factor. It is known as cleavage polyadenylation stimulation factor. Okay, and it recognizes a sequence which is known as AAA. Okay, then there is other one factor which is known as CSTF, which is known as cleavage stimulation factor. It is known as cleavage stimulation factor. It recognizes the GU-rich sequence. It recognizes GU-rich sequence. These are two types of enzymes which are present, CPSF and CSTF. What does the CPSF does? It recognizes the sequence AAA, which is present before the cleavage site, and CSTF, it is a cleavage stimulation factor. It recognizes GU-rich sequence and it binds. Then there is the RNA cleavage, and the enzyme CSTF is being removed because the RNA is being degraded, or we can tell that there is a cleavage of the RNA and this enzyme is being removed. But here the binds the CPSF, which is known as cleavage polyadenylation stimulation factor, because now the cleavage has been completed but the polyadenylation is going to take place. Okay, so here the another enzyme comes and bind, which is known as poly-A polymerase. It is also known as PAP. What is the function of poly-A polymerase? It is a polymerase, that means it will add the nucleotides. It is poly-A polymerase, that means it will add multiple adenine by the help of the polymerase activity. After that, there is a binding of poly-A binding protein. Here we can see that PAP has binded over here, due to which there will be formation of poly-A. Long tail of poly-A will be formed. Here we can see that here there is long tail of poly-A. So for preventing this adenine or the stretch of adenine, there is another protein which is binding, which is known as poly-A binding protein. We can be very clear with this diagram. Here we can see that there is a factor which is known as AAA, which is present upstream of the cleavage factor, which is being recognized by CPSF, which is known as cleavage polyadenylation stimulation factor. It is a stimulation factor which is responsible for cleavage as well as for polyadenylation. There is another one enzyme which is known as CSTF, which is known as cleavage stimulation factor. It is only responsible for cleavage, which is being attached to the GU-rich sequence. Okay, so what happens after that? It will form a loop-like structure, or it will form a fold, due to which there will be interaction of these two enzymes, which is known as CPSF and CSTF. Okay, after that it will help in recognition of CF1 site, CF1 and CF2. It will help in the recognition of this site which is responsible for the cutting, and it will be recognized by CF1 and CF2, and there will be a cut in this portion. Okay, this is the process of cleavage. Then another enzyme will come and bind here, which is known as PAP. PAP will come and bind here, which is a polymerase. It will help in the polyadenylation of this adenine, and for preventing this or for stabilizing this adenine, there is requirement of an enzyme which is known as PAB2 and PAB1. Okay, this is how the polyadenylation is taking place. Okay,